Hey there, welcome back. Get ready for molecular and cellular biology terms L through O. Stay tuned. Welcome to Science is You, a new channel dedicated to making real biotechnology research enjoyable and accessible to everyone. To support this mission, please subscribe and spread the word. Thanks for watching. L. Ligase. Ligase is a type of protein that joins two things together. DNA ligase, for example, joins two ends of DNA together. DNA ligases are critical both for repairing damaged DNA as well as part of the DNA replication process that happens when cells reproduce by dividing. Microarray. Microarrays are used for seeing what parts of the DNA a cell is reading in preparation for turning them into proteins. Remember that the parts of the DNA called genes are first read or transcribed into an intermediate messenger molecule called RNA. Then the RNA further is translated into a protein. In this way, the cell passes information through various forms to achieve its final goal of making proteins that enable the cell to do different things. Transcription, the process of copying portions of the DNA into RNA, is a valuable process to study in order to learn how the cell changes, adapts, and takes on different forms in the body. If you recall the hybridization term from earlier in this video series, two strands of DNA or RNA that can bind to each other are called complementary, sort of like pieces of Velcro. We can take advantage of this in the lab to detect specific sequences of DNA we are interested in. For example, say we have two samples of cells we are studying. One group of cells are healthy, and the other we treated with a chemical that we think might cause cells to become cancerous. To look at what changes the chemical may have caused, we can extract RNA from both groups of cells. But RNA in the lab is unstable. Similar to how the cell copies DNA into RNA, in the lab, we can take RNA and copy it back over into DNA so that it's more stable and we can use it more easily. A microarray is basically a slide containing hundreds or even thousands of DNA sequences that are complementary to sequences for genes we are interested in studying. These are arranged in the slide in a grid-like pattern or array. By attaching a type of dye to our samples, we can see on the grid where our samples attach and about how many of each type of sequence is present. We can then infer whether the cells we treated with chemical are making more or less of certain proteins. Nuclease. Nucleases are special proteins that function to destroy nucleic acids like DNA and RNA. Wait, why would the cell make proteins that destroy its genetic material? Nucleases have many varied roles in the cell, including helping with the DNA repair process, recognizing and destroying foreign microbial DNA and RNA, and also function in apoptosis, a mechanism where a cell that has become damaged or cancerous actually destroys itself, keeping the surrounding cells and tissues healthy. Oncogene. An oncogene refers to a gene that has become mutated and has the potential to cause cancer. A gene that plays a critical role in cellular growth, division, or survival is called a proto-oncogene. Once mutated, this gene is referred to as an oncogene, since changes in genes encoding for proteins that are critical for the cellular growth and survival balance might cause the cell to start growing and dividing uncontrollably, causing cancer. Think of it like the brake on your car malfunctioning. Without it, things can get out of hand. There you have it, molecular and cellular biology terms L through O. If you enjoyed this video, please subscribe and stay tuned for the next video in this series, and remember,